My name is Kasthuri Venkateswaran. People call me Venkat. I'm affiliated to NASA Jet Propulsion Laboratory, administered by Caltech. If anybody interested to contact me, please use the following email that I have given here. Today I'm going to talk about the integrated microbial monitoring system of the crew and engineering systems for future human habitats. Today I'm going to talk two major things. One is with respect to the planetary protection. The other one is human habitat. So, what do we need for future exploration with reference to microbial monitoring? This will help to assure the evolving standards for planetary protection with reference to human exploration of other celestial bodies. This includes both forward and back contamination related issues. The second major thing is when we have human in perspective, we cannot have a clean habitat because we are walking fermenters. We constantly shed microbes. So a sound technical basis to determine whether the inadvertent shedding of these biocontaminants from human explorers can be minimized to such a degree that the search for life can continue in an unobstructed, meaningful manner. The second one is the habitat themselves. Microbes might corrode these things. So, with reference to this, it is called biocorrosion, can be mitigated. So, what are the technology gaps? We need integrated system technologies that protect human life. When I said integrated system, there are a couple of scenarios which I told you before. One is the planetary protection scenario. So the problem, we have to address planetary protection policies for forward, moon, Mars, beyond, and back, Earth contamination. So this is one aspect. That is a problem. That's what the planetary protection officer do um, you know, to contain any kind of forward contamination and also make sure that back contaminations are not happening. What is the state of the art now? Current PP requirements address only robotic missions to Mars. For human mission, it is still in a conceptual stage. With reference to the technology or capability needs, there are so many things here you can think about it. Waste management, microbial dispersal outside, cleanliness inside, microbial monitoring, biobarrier, cleaning, sterilization, and how do you control the unwanted microbes. So we need, these are the needs. Do we have the appropriate capability? We don't know. With reference to forward contamination, like moon, category 1 and 2, containing the dispersal microbes and Mars, category 4. So these are the two scenarios that you can think about the forward contamination. So countermeasures for microbial proliferation, containment, and control are needed. With reference to back contamination, moon, there is no restriction, so no problem. And for Mars, restricted entry back. So we need to have appropriate sample receiving facility, adopt Mars, sample return project technologies to address these planetary protection policies. So what do we have? We don't have a TRL-9 technologies to meet these PP policies for manned mission to Moon, Mars, and beyond. So those has to be appropriately studied. So the microbial monitoring system characteristics are given here. Some of them are, pointed out here. The MMS should be an integrated engineering system for sampling to detection and has to be validated in a terrestrial Mars analog environment. In addition, it should estimate viable bioburden present within fluid that are being used by astronauts, surface that are being used to live in there, and the air that are being breathed by the astronauts from this, using some sort of simulated lunar and Mars habitat. And also the dispersal of microbes from the human habitation has to be checked because This will be a huge issue when you are looking for life in other planets. So integrated microbial monitoring system should be implemented and tested before taking it anywhere. As we know, something like lunar and Mars crewed flight activities should be, you know, happen before we even think about taking it to the other planet. So scenario number two. This is mostly about human living. The first scenario I was talking about it there is mostly robotic missions collecting samples from other planets. And this scenario is about the human involvement having a you know um, habitat where they live in. So one most important thing is biocorrosion, then biofouling, and then biofilm formation. So what are the problems? Controlling and monitoring microbial proliferation and its influence on crew health and structural integrity of habitat system. The state of the art is ground-based solutions are available, but no autonomous microbial monitoring system and microbial monitoring and control systems are available. In the past, LOCAD was used to measure dead and alive, but not metabolically active microbes. So similar to that simple, smaller scale, handheld microbial monitoring system might be helpful. The gaps that are identified here is to differentiate dead from alive to measure metabolically active microbes because the the metabolically active microbes are the one are problematic to crew health. Second is cleanliness of structural integrity. If we are not able to have a clean structure, then biocorrosion will happen. And also more importantly, not all bugs has to be cleared. Medically important microbes has to be appropriately uh, you know measured, and countermeasure has to be developed for that because that is directly related to the crew health. So what are the technologies needed? Mitigation technologies, something like microbial monitoring technologies like IMMS, and European Space Agency are looking into things like called MIDAS, are the one already flown like LOCAT 2. In addition to this, we need artificial intelligence for autonomous deployment because very less crew involvement is required. An artificial intelligence for autonomous deployment will help the crew to get an intelligent decision made. The scope here, the mitigation of biocorrosion, biodeterioration, biofouling issues. So these has to be tackled. Issues pertaining to the microbes metal interaction is another big issue that has to be studied very well. And also we have to have a complete characterization of microbial diversity associated to the closed system like what we are doing right now for International Space Station. So it should be flight deliverable and a flight proven to the level of TRL 9 with reference to maintenance and microbial monitoring system because crew will not be able to do this. This should be autonomous. So technology is required to control the harmful impact of microbial corrosion. We have to plan in mitigating these contamination and also facilitate system cleaning, system sterilization, and maintenance by incorporating and adapting precision and automated, semi-automated cleaning. We need to develop sterilization technology geared towards mitigating cyclic human recontamination, including dry heat for the hardware. We can use radiation for the both hardware and liquid. Vapor hydrogen peroxide is also useful for hardware and habitat, um, decontamination. Filtration are normally used for liquids, and also you can use it for the air to filter to pump into the habitat where the crew live in. Wet heat system, like not only the dry heat. Sometimes you may need to have a wet heat system for sterilizing the liquid. And Extreme cases, you need to fumigate also the whole habitat. So one more thing, not only the developing this kind of technology, but also a kind of controlling using a kind of biobarrier, where we all know right now, a lot of people are very familiar with coronavirus biobarrier now. So likewise, we need to develop some biobarrier technologies and other secondary human-driven contamination avoidance technologies to prevent biocorrosion, biofilm formation, and to maintain clean, sterile system. So controlling the harmful impact of microbial corrosion. First of all, we need to compile the comprehensive inventory of biocorrosive microbial communities by measuring the crew microbiome, what microbes are associated with the crew, as well as with the environment they dwell with. Already, space biology has started microbial tracking program. Now we are in 9 years program right now. That gives you some information about what is associated with this kind of environmental microbiome. But there are a lot more of studies we need to work with the crew microbiome. And also develop and demonstrate the flight readiness of an artificial intelligence biocorrosion microbial sensing system that can provide autonomous, semi-autonomous microbial contamination mitigation and also control such as system cleaning and sterilization. This is an example about the microbial succession in space that is not different from what we are seeing in Earth. First of all, with metals and other surfaces, microbes crawl in and colonize, including spacecraft surfaces and other things. But we have a lot of spacecraft microbes that are, doing things, highly um, enhanced biofilm are being tested. And also, are there anything happening in the space? So we can take, you know, set of microbes outside this, low Earth orbit to see how the microbes are adapted in space, and then look into the molecular mechanisms that acquire. It is not only the benign microbes that are radiation resistant is a problematic, but horizontal gene transfer that actually, you know, give the genes in terms of these functional proteins to the problematic microbes like E. coli and other things, that will be a problem. Acquiring the genes that are responsible for this radiation resistance and other conditions, and then transferring them into a problematic microbes is a big issue. Then, if we are not able to do all these things right, then we will start looking, seeing this biodeterioration of materials and biofouling, which is very, very expensive. So presently what we are doing with reference to microbial monitoring is we collect samples, store them. After a couple of weeks or months, it comes to the earth. So it takes about totally from sample collection to reaching to the lab around 4 to 6 months, and then transport to JPL, transport to the any institution, and then do all the benchtop laboratory setup to see what are the tree of life here. Instead, if we have a technology to extract appropriate biomolecules of interest, which takes within a day or 8 hours, that can be appropriately measured using molecular methods like MinION and do the sequencing in space to look into the tree of life, what are the problematic microbes are there. So integrated microbial monitoring system characteristics contain three big parts. One is how do you collect samples? So for water, surface, and air, and even crew, we need a suitable sample collection system. There are carts available and should be tested. Then we need to have appropriate, processing so that the biomolecules of interest can be extracted in space. So we already work with some SBIR product. NASA is already working with SBIR product that is available and microgravity compatible. Then we already having in space MinION, Razor, and WITLAB systems that can be utilized. But the biggest issue is how you collect these samples and integrate that with the processing step. And then that further feed the biomolecules of interest into the sequencer or the PCR system that we have up there. Or in future, we can have a litmus test kind of system like ATP assay or any other system should be developed for the integrated microbial monitoring system. So this is the final slide. The gap in technology for human mission to space. Microbial detection and their mitigation system to facilitate spacecraft habitation, cleaning, sterilization, and maintenance. These are the paramount importance right now to prolong the longevity of human habitation on other planet. Second is life support process. There are a lot of recycling materials are happening in humanity out there. These will promote the proliferation and colonization of microbes, which are likely to play a role in subsequent biofouling, biocorrosion, and also pathogenesis of host organism, in this case crew, as well as plant. So in-depth understanding of whether spacecraft and crew-associated microbe can provide insight for a more efficient, low-cost, practical approach to specific challenges in space. These are the gaps, but what do we need? We need validated environmental monitoring system and control strategies are crucial to preserve acceptable microbial burden level, this is for planetary protection, in human compartments and life support components. That's a crew health related issue. And also, such a system will ensure negligible interference of false positive with life detection experiment as well, and also prevent an inadvertent exposure of humans to extraterrestrial materials. Thank you very much. Bye.